Hey guys, so we'll be going over some of the types of microbes that you guys can be expected to be tested on test day. So the first one that we have up is our Archaea bacteria, and these are similar to that of the Eubacteria with a few differences. While regular bacteria uses ester linkages in their plasma membrane, Archaea bacteria uses ester linkages, which allows for a higher bonding for the plasma membrane. And we'll get into why that's significant in a little bit. The outer layer that Archaeobacteria have is similar to that of peptidoglycan, with a few differences such as they use L amino acids instead of D amino acids, and they have a beta 1,3 instead of a beta 1,4 linkage. And they also use a different kind of acid. I'm not even going to try to butcher the name, but as you can see, the first one over here is what's used in Archaeobacteria, and this is what's used in regular Eubacteria. And all of these things allow for the Archaeobacteria to survive in harsher environments. It allows for them to have a better, it allows for the plasma membrane to hold better together, like say in extreme hot, cold, or high salinity. Alright, and they also have an ATP binding consent, abbreviated to be ABC, and this is for their nutrient uptake. Alright, now before we get into other bacteria that you guys are going to be looking at, I just wanted to mention that with Archaea bacteria, I've left out that the differences in their cell wall structure make them resistant to environmental changes, or extreme environments, I should say, but also make them resistant to things such as lysosomes and drugs such as penicillin. And now we're going to go into the differences of prokaryotes and eukaryotes. As you can see, prokaryotes are unicellular while eukaryotes are multicellular. And what that means is that with prokaryotes, all the cells are the same, while eukaryotes, it takes many different cells to make the being. And prokaryotes have no membrane-bound organelles, while eukaryotes do. So what that means is that within each individual cell, that the different aspects of the cell are compartmentalized, while in prokaryotes, it's just kind of all floating around. And we have linear DNA, as normally see with the double helix, and prokaryotes have a circular DNA, and they divide by binary fission, which means that 1 becomes 2, that 2 becomes 4, that 4 becomes 8. While eukaryotic cells go through mitosis, which is through all our cells except for our gametes, which will then go through meiosis. And the flagella that we have are composed of two different things. Prokaryotes have flagellin while we have tubulin. And the ribosomes that we use to make our proteins are even different, where ribosomes have a 30S and a 50S subunit. When they join together, make a 70S subunit, while eukaryotes have a 60S and a 40S that when they come together make an 80S subunit. And prokaryotes have no mitochondria while eukaryotes do. And in order to compensate for that, prokaryotes make their energy within their cell wall. Now getting into prokaryotic classification. We're going to be looking at clone and strain. Now, you guys have had the lab where you guys have streaked for isolated colonies. Your clones are going to be all the bacteria that are in one isolated colony, while your strains are going to be the different variations of that same bacteria, and those are going to be all the different bacteria in neighboring colonies. So one more time, the clones are all the bacteria in one given colony, because they're all the exact same one, while the strain is the different bacteria that are in the neighboring colonies. Alright, the other microbes that you'll be looking at are fungi, and these can be single and multicellular. They have membrane-bound DNA, but they do not do photosynthesis. Although they kind of look like plants, they are not plants. So photosynthesis in fungi is a no-no. And they receive their nutrients from the environment. If this is a bit confusing for you, look back at your nitrogen cycle and you can see that they decompose some of the stuff that is retaken up by plants. Alright, next up we have our protozoans. Our protozoans are unicellular beings, and they move by pseudopods, flagella, or cilia. And this is very obvious, like when you guys use those wet slides for your guys', I think it was like probably like your first or second day. That's how they move around, is by using those pseudopods, those little feet. And pellicles are supporting cells that help them retain their overall shape. And they help out as far as the food chain goes. They're a big source of food for the microinvertebrates. And they're consumers as decomposers. And what that means is that they eat material that nothing else will. So they'll break down stuff like dead material or stuff that's no longer viable as a food source for other species, and they consume that. So they prey on things that are non-living, and they go over and they divide by binary fission, as well as bacteria, and they survive in harsher conditions. Now, the last microbe that we're going to look at are parasites, and these are eukaryotic beings, and they're classified as helminths. Now, what they do is they weaken the host overall by stealing the nutrients from them. So it's kind of like they're siphoning out the nutrients before it can be useful for the host. And with that, I hope you guys found this helpful. Again, you guys don't need to be an expert on any of these things. This is just a brief general overview of what you guys are expected to take a look at in the foreseeable future. So for this midterm, don't worry about being an expert on any of these things. Just understand like the few things that they have in common and the few things that make them different from one another. If you have any questions about this or anything else, feel free to shoot me an email. You guys have a good one.